Well, Uroš, first of all, thanks a lot for the invitation. It has been, a great pleasure to be here, even if it is only virtually at this stage, but I'm sure that there will be indeed occasions for a meeting also in presence. So I start with introducing this one. Okay, may I ask you whether you can see the PowerPoint? Yes, okay, great. So, well, as it was mentioned by Uroš, today I will introduce some of the research topics that I've been working on in the past years, which actually are, focusing on the applications, but first of all, on the developments of methodologies to perform functional connectivity analysis using electroencephalography, in particular high-density electroencephalography, to investigate brain networks. So what you are going to see here, it is actually something that we will probably, at least we aim to use also for our latest bed rest acquisition campaign that has been performed this year in Copper. So yeah, I will shortly introduce this concept, and then in the second presentation, I will show you this software, NET, Non-Invasive Electrophysiology Toolbox, that has been developed in the past 10 years with my former and current research group also at the KU Leuven, together with a large group of engineers working in the field of neuroscience. So first of all, I will introduce what is actually functional connectivity in the brain. In particular, during my career, I've been focusing on functional connectivity at rest. So means that we are not studying the brain in a condition of active task, but in a moment of mind wandering. So people, when we perform resting state acquisition, we ask them to not think about anything in particular and just to relax. And basically, with the concept of functional connectivity, we look for interactions in between signals from different brain areas. So functional connectivity is defined as the statistical dependence between the activity generated in different cortical regions. So for example, in this case, you can see from the signal that we record from the region of interest in red, we have a signal, we have another one from the region in green. And so we can see that there are similarities between these two signals. And so these signals, they will show a high correlation, which means that when we have a high correlation, we can define a brain network. So two areas that communicate in a privileged manner. For example, the signal from the region of interest in blue doesn't have a strong correlation, therefore doesn't belong to a specific brain network. The thing is that these brain networks and these resting state networks, more specifically, have been studied a lot in the past years. However, they've been investigated using functional magnetic resonance imaging, and which allows somehow to detect the hemodynamic changes that are happening in different brain areas. Something about the resting state networks is that these areas are connected to each other. We have different brain networks that recapitulate a certain behavioral function. Therefore, we can define attentive networks, sensory motor networks, or auditory networks. So means basically that these regions that are involved during an active task, they're also strongly connected when the brain is at rest. So even in absence of an explicit task, these regions are connected. So it seems like that they represent a sort of baseline of human brain processes supporting cognitive and motor action. And there has been a great interest about these brain networks also to identify correlates with another technique that has been used for studying brain function, which is electroencephalography, for example. As I was mentioning before, fMRI allows to detect the hemodynamic activity, while EEG allows to detect the electrical activity. And so back in 2007, researchers wonder whether there was a link between these hemodynamic activity and electrical activity. Therefore, they correlated the time courses associated with these fMRI-derived brain networks and the power of the EEG signal. And so you can see that every network here, for example, we have the default mode network, here a dorsal attention network, here, for example, a sensory motor. You can see that we have different values of correlation with the different frequency bands, which suggests that depending on the network, somehow the communication is supported by specific brain rhythms. Therefore, the alpha and beta GAM are quite shared in different networks. We can see that the motor network, for example, is characterized by the beta band, which is something that we could also expect, knowing that when we have an active task, also we have an increase or something happening at least in the beta band. So it seems that there is a strong relationship between resting state connectivity and also activity during active task. However, there are still many aspects that should be explored in this direction. However, with this study, it was somehow suggested that the different brain networks can be expressed also in terms of electrophysiology. Therefore, there's been a great interest and effort trying to develop some methods for extracting these resting state networks also using neurophysiological techniques. This one has happened in 2011 using magnetoencephalography, the MEG. And you can see that the authors could achieve somehow something similar to what was extracted with fMRI. So looking that there is actually a spatial correspondence between the different resting state networks acquired with the two techniques. But how about EEG? Well, let's say that to do that, it has been a more delicate issues because more methodological developments were required, both in terms of hardware and software, which is what we will see today. So indeed, the question is, at that point, could we actually detect the resting state networks also using EEG? Well, that's the spoiler. Yes, we can now. However, it has been a long journey because EEG notably has been investigated at the scalp level in the sensor space. So we had our electrodes in the head and we were recording different signals. And these signals, usually we could either look at the temporal domain or at the frequency domain with all the separation in the different brain rhythms that we all know with the delta, theta, alpha, beta, and gamma bands. And actually, this one has been largely explored for decades. So we know that, I mean, in correspondence of a change of cognitive state, we have also a change in the neural activity. For example, if we consider in the time frequency domain, the simple transition from having the eyes open and the eyes closed, we know, everyone knows somehow, this has been detected back in the 30s, last centuries, that there is an increase in the alpha power when the eyes are closed. But all these analyses, they are linked somehow to the sensor space, and most importantly, were linked to low density EEG systems, like the one that you see here. However, to extract the brain network somehow, we need to increase also the spatial resolution of our EEG systems. And for this reason, thanks to the introduction of high-density EEG systems, which have a larger number of electrodes, it is possible to perform an accurate localization of the sources of the EEG. And therefore, we can work then in the volume of the brain somehow to extract some information from our data. So here, for example, you can see a 128 system, which is kind of similar with the one that has been used in Copper for the acquisition campaign. So it means that with the data that were acquired recently, we can perform accurate source localization. But let's have a look more in detail. So basically, what we aim to do was to move from the sensor space. So here we can see a topological map. I think this one is after the performance of a go-no-go task. So we have some activations in the attentive region somehow. At least here we can say not attentive region, but we can see over the electrodes over a certain brain region. And when we perform the source localization, we can actually see that the major activation is in regions belonging to the dorsal attention network. So we can really have a more detailed information on where actually the signal that we detect has been generated from. And so for going from the sensor space to the source space, we had to develop a workflow to perform EEG source localization. And this workflow includes different steps. So a step of data processing in which we analyze our high-density EEG data in the sensor space. And so we perform the typical data pre-processing that is performed to lead EEG analysis. So including bad channel reparation, filtering, ICA, denoising, and artifact removal, and re-referencing. But together with this, we also have to define a volume conduction model that basically tells us how the neural signal that has been generated in a certain brain area will move towards the surface of the head so that can be detected from our electrodes. So it is very important to model this path that the current has to flow from the inner part of the brain towards the outside. And for this reason, we need the information about the geometry of our head, so about the anatomy of our subject. We need the information about the position of the electrodes, because the signal that we will record here will be different from the one recorded here. And so it's important to have an accurate information also of this electrode position. And then we will combine this information about the head geometry and the electrode position to build a head model. Most importantly, another aspect that has to be taken into account is the segmentation of the head, that basically starting from the individual structural MR image will define some compartments in the brain. And each of these compartments will correspond to a specific brain and head tissue. And so we can define the geometry of these head tissues, these compartments. And together with this, we can assign specific conductivity values. So we need to assign electrical properties to these tissues. Therefore, we can know how easy will be the flow of current in this specific tissue. Then combining our clean EEG data and the head model that we built with a mathematical transformation, somehow we can actually move from the sensor to the source space. And so we can reconstruct the brain activity inside the brain. At this point, the maps that we can achieve and also the way the activity is displayed is actually very similar to what we could achieve with fMRI. Also, in this case, we talk about voxels, and therefore we increase the number of our signal. If in the sensor space we had 128 or 256 channels, then when we move to the source space, we might have up to 3,000, 4,000 voxels. And this one is similar to what actually is also in a fMRI data. This doesn't mean that the resolution is the same. Of course, we could improve with these methodologies the spatial resolution of EEG. Now we are up to 6 millimeters voxels. Still is below compared to the one that we can achieve with fMRI, but it is still very interesting, and you will see what we can achieve. So once that we have our sources reconstructed, we can perform connectivity analysis. There are majorly two approaches that could be used. One is ICA-based, therefore independent component analysis. I'm sure that you heard about this concept, at least for the artifact removal part. And then the idea is somehow that starting from a set of signals, basically this set of signal is composed by a linear combination of different contribution of different spatial patterns. And so we can see that there is a spatial pattern that can be extracted, and it is a spatial pattern that it is independent from all the other ones that are extracted. And together with this spatial pattern, we also have a time course that is associated to it. So by using the ICA to perform connectivity analysis, we basically ask the data to tell us what is the typical patterns that are inside and which ones are independent. And so some of these patterns, they might be ascribable to brain networks, while other ones, they might be simply noise. But the important thing is that this approach is data-driven, so it is great for exploratory analysis. While another approach that could be used, well, we will see it afterwards, it is the seed-based. In that case, we can define hypotheses a priori. Therefore, it's not data-driven, but might be more interesting to test our working hypothesis. Here, you can see some examples of what we can achieve with the ICA analysis using the high-density EEG. So in this case, so we have the reference of functional magnetic resonance imaging networks for the default mode, for example, and the dorsal attention. So you can recognize typical regions that are involved in the default mode network, and here also the typical W shape of the dorsal attention. And here you can see the results of a group analysis of 20 subjects, I would say, using the independent component analysis approach also on the source localized EEG data. And most importantly, you can see that here there is written T and S, which stands for temporal and spatial ICA, respectively. Basically, this defines the way that we orient our matrix of signals that we give as an input to the algorithm. And let's say that at first we thought that temporal ICA could have been, let's say, more accurate because the independent component analysis algorithm works somehow better whether there is a large amount of data. So we know that with EEG we have a high temporal resolution, therefore we have many time points. So it was somehow, well, made sense somehow to try to expect that the temporal ICA could lead to more accurate results. However, we found out also that the spatial ICA lead to excellent results, and actually they resemble even more the spatial patterns of the fMRI. And this could be explained by the fact that, as I was mentioning before, we don't have a small number of inputs because we don't have anymore a number of channels, but we have a number of voxels. So we already moved to something that is actually more similar to the fMRI as well. But with the EEG, we don't have only the spatial resolution of the functional MRI, but we also have the information about the frequencies that are involved. Therefore, we can see actually, as it was suggested in the previous studies that I showed, that the brain networks are characterized by specific brain rhythms. Therefore, with the EEG, we could reconstruct fully this information. And so you can see using the ICA that we could decompose the signal in a certain way that allows to extract the brain networks. And these brain networks, their topology, their spatial organization, depends on the frequency band that we consider. So for example, in alpha band, you can see that are involved all the regions that belong to the default mode network. Here we are showing the default mode network in different frequency bands. And while this doesn't occur for other frequency bands, for example, for theta, we miss the right angular gyrus, but we still have the left one. We have the posterior cingulate cortex and the medial prefrontal cortex. For delta, for example, the connectivity is stronger only in the posterior cingulate cortex and in the right angular gyrus. And this change also for the higher frequency bands in which we don't have strong connectivity, strong connections in the higher frequency bands for beta and gamma, where we don't have the medial prefrontal cortex, for example. So at this point, we could, starting from this data-driven approach, we could go to an hypothesis-driven approach, such as the seed-based connectivity. So, for example, we could consider ICA extracted network. Here we can see the example of the ventral attention. And we see that this network is composed by several regions somehow. The activity of these regions is strongly connected between each other. And so we might be interested in seeing how the connectivity changes between the signal in these specific regions belonging to the network and the signals from the rest of the brain. So for example, in this case, we can see that by setting a seed in the right temporal parietal junction, TPJ, we can see that actually the connections of these regions with the rest of the brain resemble the spatial pattern of the network itself. Therefore, this region doesn't communicate with other regions external of the network. However, this other region, the inferior frontal gyrus, this one, yes, it communicates instead also with regions that doesn't belong to the network itself. So it is possible actually to explore not only the spatial patterns, but also the interactions of these regions with the rest of the brain. Here is another example with a sensory motor network, which might be of interest also considering the mobile application of the latest research campaign. And also in this case, we can see that there is a stronger connection with certain region. So here is the approach again of the workflow to perform the seed-based analysis. So we start again from the source reconstructed EEG signal, and we define somehow the power of the signal for each voxel in the brain. And so we are able to display this information, this connectivity information in two different fashions. In one case, we can perform and define connectivity matrices frequency by frequency. So we can have a very parcelized information in the frequency domain. And we can also generate maps for showing, for displaying how the signal from a certain region of interest correlates with the other brain regions. So this is actually, well, more visually saying what I was saying before. So we consider our time course from one seed. We see how this correlates with the rest of the brain. We see that these areas are majorly correlated with the signal from this seed. And we can define the region of interest in the brain to check also the correlation values in the shape of connectivity matrices. And a connectivity matrix is actually a correlation matrix, to make it very simple somehow. So when it is red, in this case, we have a higher value of correlation. Therefore, it is maximum in the main diagonal, and it is lower in the outside of the main diagonal, showing the correlation between signals from different brain regions. And so in this case, again, we can see that there are differences depending on the frequency band that we consider. So for example, again, in the alpha frequency band, we can see that the reconstruction of the default mode network includes all the regions. So it means that those regions in the network are strongly connected to each other, while this doesn't occur for the other frequency bands. For example, we can see that here is weakly connected the signal from the medial prefrontal cortex with the posterior regions of the network. And these change for the higher frequencies where we can see that there is a higher correlation. If we change, for example, well, the seed, we will have different spatial pattern again, so that we can have truly a frequency-specific information on how the different brain regions interact with each other. So at this point, well, we show somehow that we are able to reconstruct resting state networks that are similar to the one achieved with the fMRI. So here you can see the one with fMRI and the one extracted using the high-density EEG recording. So this is actually, well, a nice achievement. And so at this point, we can actually think about different applications. And one thing that I've been somehow focusing in the past year is actually oriented also to investigate somehow which are the functional changes that might occur in the brain of the astronauts. There is still little that is known about this, but the little that we know is about fMRI recording. You can imagine, however, how how cool but also useful could be actually to perform resting state network mapping of the human brain in space and not only on Earth. And of course, there are many steps that should be done in this direction. And would be of great interest because indeed on Earth, we are doing our daily activities in a condition of normal gravity, while in space it is in condition of microgravity, which is a gravity close to zero. So therefore, we can expect actually that the brain would work in a different way. The human body works in a different way, and the effect of microgravity strongly affects actually human physiology. So while we know that there are changes at the behavioral level, at the motor level, and at the attention level, we still don't know what is the true correlate of these deficits somehow of impairment that happens. We don't know what is the correlates with the brain changes. Still, there is some work that has been done in this direction with using some spaceflight analogs that allows to reproduce some physiological adaptations that happens in space, and therefore allows on Earth to explore somehow what is the effect of microgravity. And specifically, we talk about simulated microgravity. This one can be somehow replicated and investigated by using different approaches, such as the parabolic flight, for example, or the head down and the traditional bed rest condition, which is actually also explored in Cooper. And so there are some little things that we know about it. So for example, for the parabolic flight, we have, we are able, researchers are able to reproduce a gravity zero condition. And it has been seen that with functional magnetic resonance imaging performed before and after parabolic flight campaign, actually there are some changes that occur that persist at the functional level in regions that are part of the default mode network and attentive networks. So here, for example, we can see posterior cingulate cortex and angular gyrus. Which is something actually interesting because the default mode network, it is somehow a mediator between the active brain and the passive brain. So in the functioning, it is an active component in the functioning of the brain when it is mediating the information coming from the external world and the inner world inside our bodies. So it is very interesting that it is the network showing differences also with such a short exposure to simulated microgravity. And also, another model that has been largely used is the head-down bed-rest model, where somehow some physiological adaptations, such as the fluid shifts towards the upper part of the body, is induced by this specific body posture. And also in this case, it has been identified a change in the cortical excitability. So there are some changes in the way the brain exchanges information with the rest of the body. And for example, this cortical excitability has increased for the brain regions involved in the leg movements, which is the one more strongly impaired by prolonged bed rest, while it remains quite similar for the hand representation in the human brain. So also here, there are some neuroimaging data that show that there is also a topographical shift, so that there are actually different regions that are involved. So it seems like that the brain take advantage of its plasticity to recruit novel regions to perform a certain task. And let's say that one aspect that we found to be relevant somehow before going to a true head-down bed rest is actually to check whether with our high-density EEG recording, we are able to detect more subtle differences in brain connectivity. And to this end, we aim to investigate, therefore, whether there are changes in functional connectivity, not only in a strong head-down bed rest condition, but also in a simple transition from a standing position, from a sitting upright position and a supine position. This is actually a very big advantage of the EEG systems because it allows indeed to record online brain activity in different body position. This is something that cannot be done with fMRI, where of course the subjects need to lie in the scanner in a supine position. And while with the fMRI we have the great advantage of a very interesting spatial resolution, there are many other aspects that instead lack in the MRI technique, but instead are present in the electroencephalography systems. Especially, we have a high temporal resolution, we have a direct measure of brain activity, and most importantly for this topic, it is small and portable, so it can be brought to the bed of the patient, for example, and can be used in different body positions. And with what I show you before, also the spatial resolution, it has increased largely with the high-density EEG recordings. Therefore, we are somehow ready to test this hypothesis and see whether there are differences in functional connectivity linked to the different body posture. So this is what we did somehow. There is some literature about the effect of body posture on brain activity. But I highlight that it is brain activity. Nobody could so far test this on brain connectivity because there was this lack of methodological issues. And still there are several things that we know about the effect of body posture on brain activity. We know that the pain sensitivity decreases, that the brain lateralization decreases, and there are some changes at rest of the power. So this has been an estimate of the power at the scalp level, so we are still at the sensor level. And it has been reported that there is an increase for the lower frequency bands, for the power of the lower frequency bands in the bed rest condition, and a decrease of the higher frequency bands, the beta and gamma. And an interesting aspect also that is worth be mentioned is that this bed rest analysis somehow allows to manipulate somehow the brain activity, and therefore we expect also connectivity. This is what we are going to explore also with the latest campaign in copper. Here, for example, in this study, you can see at time 0, the blue and the red column, they represent the sitting position and the bed rest condition, respectively. And the blue one represents the control group, in which for each time point, the recording has always been performed in the sitting position, while the red one represents the group that will undergo the bed rest. And so we can see that at the baseline, they are all in a sitting position. Then there is a decrease of beta and gamma power when the group is in the bed rest condition. This is maintained during this prolonged bed rest. And then in the moment that the people of the red group gets back to the sitting position condition, there is again a restore of the beta and gamma power as it was at the baseline level. So it means that these changes in connectivity, not in connectivity, but in activity, in power, in this case, occur shortly after. Then they stay stable a long time, and then they are restored when the position gets back to normal. one aspect that I need to specify is that the time scale of this experiment is very short. So here, time T1 and T2 are just after 1 hour and 2 hours following the bed rest condition. So already you can see that it sounds very interesting, thinking that in our case now with the new acquired data, the time frame is not hours, but it is days and weeks. So it's going to be very interesting to see what happens. However, in this case, this experiment that I've been performing at the University of Padua, we included 18 young subjects that underwent a high-density EEG recording for 2 times with 2 resting state recordings, and they were performed in one case, one resting state in the sitting position and the other one in the bed rest. But in this case, we were truly measuring a transition, because people, they were, asked to stay in the resting state position condition for 10 minutes. Then there has been 10 minutes of transition where they changed the body posture. And then we had the second resting state recording. Of course, the 2 conditions, they were counterbalanced across subjects. And then together to this, we recorded not only the high-density EEG, but also the information about the electrode position and the individual structural MRI to build the head model that I told you about, and to perform, therefore, the accurate source analysis of the EEG and acid-based connectivity analysis, where we investigated different resting state networks in different frequency bands. So here is the workflow for EEG source reconstruction with the pre-processing that has been performed automatically using our software, and also the head model creation and the reconstruction of the brain activity inside the brain. And so at this point, we use the seed-based approach to perform this analysis. And therefore, we could extract connectivity matrices for each frequency band. So here we can see in the row and the column the different networks that were involved. So the default mode network, dorsal attention, ventral attention, language network, and somatomotor and visual network. Those ones, we explored them already in previous studies, and so we use them as a reference. And the investigation that we performed included to study what was the connectivity within a network, so inside a certain network, and between network. So what is the connectivity between the nodes inside a certain network and all the other nodes of the other networks? I spent one little word about the within network connectivity. So in this case, what you see in the main diagonal, basically, in this case, for example, the default mode network, here we show the average connectivity between the nodes belonging to the network. So for the default mode, for example, we consider the signal from 4 regions. That includes the medial prefrontal cortex, the posterior cingulate cortex, and the right and left angular gyri. So we had a look, we defined the region of interest in these positions. So we had, let's say, 4 signals that were detected from those regions. And we look for the correlation between the signals that were recorded from each of these nodes. So we had a certain amount of correlation values. And what you see here is the average correlation values between these nodes belonging to this specific network. I hope this is clear. The concept, this has been done for all the other networks that were including different nodes belonging to a certain network. And then what you see for the between network here represent, for example, in this case, default mode network and dorsal attention, represents the average connectivity values between all the nodes of the default mode network with all the nodes of the dorsal attention. And this has been done for all the nodes, for all the networks, for all the frequency bands. So in our case, we included some extra networks, not only the one that I mentioned before that we already investigated in our previous study, But we also included 3 extra networks. One is the singulo-insular network and the left frontoparietal and right frontoparietal network. And let's say that the rationale for this was that these 3 networks, they represent evolutionary novel networks. What I mean with evolutionary novel, I mean that these networks, they were not identified, detected in non-human primates. So these networks, they identify uniquely the fact that we are human beings. And so I decided to include these networks for 2 regions somehow, 2 reasons, sorry. That basically I thought that this one, they could be also associated not only to the fact that we have, let's say, specific higher cognitive functions that are commonly associated with the left and right frontoparietal networks, but also that we, compared to our ancestors, we have a different body posture in the daily activity that we perform. So I thought it could have been nice, interesting to explore also this aspect. And so what we extracted were basically connectivity matrices for each condition, for the bed rest and the sitting position. And here we can see from these matrices, maybe I hope it is clear enough, there are some diamonds that basically represents whether there are significant connections between this pair of networks. And we can see, so visually, for example, that in the bed rest position, there are fewer significant connections compared to the sitting position. Here we can visualize it also in another way. What you see here basically represents in blue the bed rest condition, while in red the sitting position. And here we counted the number of significant connections that we had from our connectivity matrices. And so visually you can see that for the sitting position, we have a larger number of significant connection, especially for the delta band, the beta, and the gamma band, while the situation is quite similar between theta and alpha band. So this suggests that the major differences in connectivity in overall are for the delta, beta, and gamma band, while it is similar for theta and alpha. But we look also for, we tested also for significant differences between the 2 conditions. And so here we can see that it emerges actually that in the bed rest condition, there is an overall lower connectivity compared to the sitting position. And we can see that there are specific networks that are involved in this lower connectivity, more than others. Also, we could check for, let's say, the ratio between within a network connectivity and between network connectivity. So in case of, we perform a t-test in this case, between within and between network connectivity. And so in case that we have the red, somehow it means that the connectivity inside a network is higher compared to the connectivity that this network has to the rest of the brain. So it means that the network is more segregated compared to the rest of the brain. While in case of blue, it means that the between network connectivity is higher. So it means that the nodes of a certain network are available to communicate with other nodes external to the network itself. And so here we can see that the situation changes depending on the body posture that we have. So there are some patterns that are similar, for example, for the default mode and dorsal attention in alpha. But for the somatomotor, for example, and the singulo-insular, there are some differences. There is like a decrease in network segregation. This one can be also represented in this way. So with the we have again our connectivity matrices, and here in the main diagonal we show as significant the times where the connectivity within network of that network is higher than the connectivity between network. And with the black circle, we can see the similarities somehow. We can see that in alpha band, this ratio is similar for both conditions, for the bed rest and the sitting position. However, we have some differences for the theta band and the beta band. So in this case, for the sitting position, theta band for the visual network and the cingulate-insular network, we have a higher within network connectivity compared to the between network connectivity, compared to the bed rest condition. And similarly happens for the beta band for the somato-motor network, which we can see that is more segregated in this condition, in the sitting position compared to the bed rest. So somehow, with this study, we provided the first electrophysiological evidence of the effect of body posture on functional connectivity at rest. And in particular, we show that the bed rest condition is characterized by a lower network segregation, which is an age-related feature. So for future studies could be tested somehow the hypothesis of accelerated aging using this approach, as we might use the bed rest condition as a model for aging processes, which again could be used also in spaceflight research using simulated microgravity. And therefore, we could see that the bed rest condition affects the way the brain communicate to each other. I mean, the different brain regions communicate to each other. So therefore, this one might have an effect also in the behavioral performance and should be tested. And this represents, therefore, a very interesting research line to explore, because the impact could be very important not only for spaceflight research, but also for better understanding aging processes and the effect also of prolonged bed rest in hospitalized patients. And so with this, we might be able to develop and test also more effective countermeasure against these cognitive and motor deficits that could be associated with aging or prolonged hospitalization. So for this study, I would like to thank also my collaborators at the KU-11, Professor Dante Mantini and Dr. Jessica Samogin, that she is finishing her postdoc there, and also my new research group that I've been joining last year with Professor Alessandro Angrilli and Chiara Spironelli at the University of Padua, and Dr. Zaira Romeo also, that support me in the realization of this research project. And of course, I thank you for your attention.
Thank you, Marco. Really nice presentation, super interesting. Also the part from the bed rest, we were discussing that yesterday already here. We do have some questions. Nina, would you start or?
Hi, can you hear me?
Yes, I can.
Yeah, thanks for the talk. Maybe one question. You showed that source localization, and I was wondering which method exactly did you use, and in which toolbox or which software did you use to analyze it?
Yeah, well, so this is something that I will tell more in detail in the next presentation. Basically, to perform the pipeline that I show you, it is somehow summarized in the net software that we will see afterwards. I already spent some words about the software. So basically, it is something that we've been working on in the past years. And the idea was somehow to optimize the different processing steps that are required to perform the source localization. And to do that, we combine different open source softwares. And we consider them, each of them, let's say, for doing the best part that are meant to be. So, for example, for the pre-processing part, we we used many functions from EEG Lab for the source localization, many functions from Fieldtrip. And together with this, also, we developed many scripts, custom scripts, somehow to perform other kind of analysis. And to answer your question more in detail, in this case, we validated the software for a different algorithm for performing the source reconstruction. And in this case, we noticed that the E-Loretta provided the more accurate results. But this is something actually that could also be further tested, because in the software in net, you can select what is the localization algorithm that you prefer to use. So there is also implemented as Loretta, the weighted minimum norm. So different approaches that could also be used.
Okay, so you used E-Loretta also for the seed-based approach?
Yes, in this case, yes. I mean, we tested it a few years ago when we were still optimizing the different steps. And a colleague of mine, Professor Chai-Ying Liu, during our doctorate, tested also the different localization approaches to perform the extraction of the brain networks. And we found out E-Loretta delivered the more reliable results.
Okay, and just the last question, when you use the seed-based analysis, you based it on some atlas, or how did you decide which voxels are in your seed, and how many voxels was there usually in a seed? Yeah.
Yes. So for this analysis that I show you, we didn't refer to personalization of the brain. On the other hand, we used a typical seed-based approach as it is performed also with fMRI. So we identify the coordinates of a certain region. And so what we give as an input to the software is the X, Y, and Z coordinate in the MNI reference system. And starting from those coordinates, we define a spherical region of interest. And in our case, we define these spheres with a radius of 6 millimeters.
Oh, it's quite large.
Yeah, kind of, but consider also the resolution of, well, this is a typical value that is used also for fMRI analysis. In the case also of EEG analysis, it is even more motivated by the fact that our spatial resolution is not as high as the one of the fMRI, but still, well, it is still good enough for, let's say, discriminating between the different brain regions.
Is there a particular region why you didn't use parcellation, but you used this spherical way of defining your seed?
Well, let's say that at first we wanted to, the approach that we used first was the ICA-based approach. So in that case, we aimed somehow to reconstruct the networks in a data-driven fashion, so without having any a priori hypothesis. Therefore, we didn't use the parcellization at that step. And let's say that for the seed-based approach, we still need, and we also would like to implement a parcellization approach. But at first, we thought to start from what we had already. So the information about the regional activity, connectivity from that we extracted with the ICA, and then using that information as a starting point to perform the seed-based analysis. So I don't know whether I clarify.
Yeah, thank you.
So it is something that we would like to do also for future developments of the software to implement the parcellization.
Great. Thank you so much. Manza?
Hi, Marco. Thank you for the talk. Can you hear me okay?
I can hear you. Yes.
Sorry, we just a second. Okay, so just a quick question. So you showed a really nice relationship between like cross methods between fMRI and EEG, and how we can use one information to get either to the seed-based analysis in the other and vice versa. I was wondering if you worked at some point with the PET scans, so with the metabolic results of the network analysis, or like yeah.
Well, we didn't have the chance yet to perform cross-validation using also the PET recordings. I can tell that it is very well known, the relationship between the resting state networks acquired with the fMRI and the metabolic activity somehow recorded with the PET. So let's say that in these cases, we could not perform a direct comparison. However, we could perform a direct comparison with the fMRI recording, because some of the results that I show you derived from data that were simultaneously recorded. So high density EEG simultaneously recorded with the fMRI data, and which somehow strengthened the quality of the approach, because it allows to reconstruct similar patterns. It would be very interesting to see also how to put in relation the electrophysiological activity and the metabolic activity of the PET. I know that actually also in Padua, they have a scanner, an MR scanner, that allows a 3-modal recording. So with using both PET, MRI, and high-density EEG. But I know also that it is very, let's say, a bit, they are not prepared yet somehow to perform this kind of analysis. So I don't know whether in Copper you have the chance of doing this. In case, I mean, I would be very interested in looking at those data. I don't have experience with PET analysis though. Do you?
No, but I was wondering, like, could you derive the networks that are associated with a specific disease, for example, the metabolic patterns, and then like take the seeds out of those networks in order to differentiate different disease stages or different subtypes of disease, which is now being showed that it can be done on the metabolic level in Parkinson's and the related motor diseases. So yeah, I'm thinking ahead a little bit about my postdoc.
I think that would be a great thing to do. I mean, this is also part of the goal that we aim to achieve with this brain network approach somehow, that to investigate actually better, also from an electrophysiological point of view, these changes in functional connectivity. So yes, the answer is yes. I mean, it would be very interesting to get the information about the seed from previous literature in PET studies, and to use it as a reference for the analysis with EEG. This is certainly could be done. I mean, consider that when we selected our seeds also, nobody did it with the EEG. A few did with the magnetoencephalography, but our reference was still the fMRI. So definitely, I mean, looking for the disruption of the network in a certain disease by selecting a specific node, a specific region of interest, this is actually what we wish could be done with this approach. So yes.
Yeah, and also there's then the trajectory from invasive to non-invasive methods, and if you could like get a reasonably good results out of the EEG, then also the clinical the clinics could benefit from the results as well.
Absolutely.
And it's easier and cheaper and everything.
Yeah, also you might use not only the information from previous literature, but eventually, I mean, now you mentioned Parkinson's disease. I imagine that you refer also to other neurodegenerative disease. However, I mean, consider also the case in which you have, let's say, a true structural damage, such as in a stroke, for example. So you might have certain regions that actually are not there anymore in the brain. Therefore, the different regions must communicate in a different way. They need to find another road, and so they can stimulate plasticity. And this one hopefully could be detected using also the high-density EEG.
Yeah. Thanks for this.
You're welcome, my pleasure.
Marco, perhaps like I do have like a question also regarding the results you've presented. So the Angrilli study from the body posture changes, right, that you shown, and you said like T0, T1, T2, which were like 1 hour, 2 hours, and then the follow-ups. So my question is the following. So we always, and we had this discussion yesterday, like, you know, in connection with the ERPs. So Torben was saying that, you know, you find in the literature, and we do the same actually, like whenever there is like an increase in amplitude, then you try to associate it to something, and then you have this vision that it should be, you know, enhanced by the training, and it should be deteriorated by the, let's say, prolonged inactivity and so on. So what we found out in many bed rest campaigns is that, you know, specifically for the muscle, for the function, for the neuromuscular junction and so on, we know that there is a deterioration, or you know, that there are negative effects of this bed rest, but we're still not sure what's happening from the brain perspective if we take this very broad view. Somehow we see that, you know, it could be like the learning effect from the cognitive test that shows then the beneficial effects at the end. So after being exposed to 14 days of bed rest, you see like better results in the end, and could be just because they performed it twice or 3 times by then. But also like from the, you know, let's say here, frequency bands that you were showing. So these very fast transitions from, let's say, seated to supine. Do you think this is the reflection of some like plasticity that occurs immediately, or it's more like, you know, adaptation of the state? So if we basically go in a supine position, then of course we, you know, react to the environment differently as we are reacting it right now because of the proprioception, because of the integration of different, you know, stimuli around. But my question is just like, how we can interpret those data, right? So it's a fast transition, you see changes, but then were they discussed in, I'm pretty sure that they were discussed in a way of like negative effects, right? Of reposition or a head-down tilt, although it was quite fast transition, as I said. And then the second question, or perhaps like a comment, was just like, the results were shown from the region of interest, or you just selected specific electrodes for those analyses, or I didn't get it from the slide, sorry.
Okay. Well, I start with this final commentary. So yes, we define a region of interest. So the data were source localized, and then for each of those networks, I recalled some coordinates from previous literature, and so I consider those ones as belonging to those networks. The studies, they were from fMRI, because we didn't have, of course, from EEG. So concerning instead the previous part of your comment and your question, actually this is something very difficult to disentangle. So at this stage, I mean, still this is an unpublished work, but I really wanted to show it to you also for indeed opening this discussion, because it is very difficult, of course, to say whether this is linked to, let's say, whether the change of connectivity is linked to a physiological change actually happening, or whether it is a change in the cognitive state somehow, because indeed they are in a more relaxed way condition, and so there might be differences. So this should be explored, and should be explored with increasing, for example, the time of bed rest, which is actually something that we did, and by recording also other physiological measurements eventually to understand whether this change in the brain connectivity is linked also to other changes in the body. Let's say that the relevant thing to me is that I don't know whether this is truly linked to the physiological changes yet. However, I find interesting that the human brain changes the way somehow it exchanges information. And what would be interesting to see is whether this change in the way it communicates has an impact also in behavior, because following somehow the hypothesis that I have also that let's say that there is a decreased segregation of the network. This is something that happens also in the elder brain, so in people in aging. So we know that when it happens that the segregation decreases, there is also an impact on cognition, and the impact is negative. There is a deficit in cognition and in the motor performance. So let's say that more than saying, I mean, the fact that something is replicated somehow with a bed rest, it doesn't mean, of course, that the brain is aging faster somehow. But the idea is that it is showing features of aging faster. So I find that this is interesting because eventually, and this one should be tested, whether the bed rest induces these changes not only in the young people but also in the older people. So in that way, actually, we could be able to see whether those changes in connectivity are linked to the actual aging or to the fact that we are inducing a aging-like change. So I think that the data acquired between August and September will help also answering this question.
Yeah.
So I don't know whether I gave a satisfactory.
Yeah, I was just like wondering, perhaps regarding the first comment or the first answer was more about the question of the analysis of the study you showed performed by EEG, if I'm not mistaken. So Angrilli, yes. So that was just.
Yeah, that one.
Yeah.
Sorry. Concerning that study, that one was performed with a low density EEG. Mm-hmm. And yes, it was referring to the sensor space analysis. So it was, I think it was with 32 electrodes, and they were analyzed at the scalp level. So the power at rest at the scalp level.
Okay. Yeah. No, so super interesting. Also, like I didn't know the study from Roberts that you showed. I have written it down just to check it out. But yeah, I now realized that my first review was basically published in 2014 and was basically summarizing the effects of electrocortical activity, or like say microgravity, hypergravity on EEG. And there was at that moment only one study from Guy Cheron from Belgium, who basically showed that in the space there is like a shift in alpha peak power from, let's say, 9.9 to 10.4 hertz, but not.
Okay, so there is an increase in the alpha peak when it is a shift towards higher frequencies.
Yeah, but in very small.
Yeah. No, well, the study from Donna Roberts about the cortical excitability, that one is actually, I find it very astonishing because, I mean, the sample size is 4 subjects. Mm. It's very, very small, but it has been a very long bed rest. I mean,
90 days, right? Yeah.
Yes, I mean, very, very long. I mean, it is crazy how convincing to me is that result, because they didn't not only recorded the neuroimaging data, but I think they also performed some TMS stimulation to actually assess truly how the neural apparatus from the brain towards the rest of the body somehow was working. So that one is actually much more convincing than any neuroimaging let's say analysis that we could perform, because of course we do perform very advanced analysis, but a TMS analysis, I mean, stimulation with the registration of the muscular evoked potential, that is something clear about the anatomy of the neural structure. So this is also something that in future studies, I think, for the bed rest should be included to perform stimulation of the brain or of periphery of the body to see how it is preserved, also the neural architecture for the peripheral.
During this break, I can share some results. We do have a publication together with Paolo Manganotti from the last bed rest campaign on the peripheral nerve conductive velocity. So I can share that study and perhaps the review. So for all the others who might be interested.
That would
perfect.
be
So do we have like any other questions here? Online, perhaps? No, I think we are perfect in time. I see now 10:15. So
amazing.
Okay, then we go for a break. So let's say how much time we have? Half an hour. Okay, is that okay with you? For you, Marco?
Yeah. So we connect again in 30 minutes maximum. Yes. Around. Okay. That's
okay. Perfect.
Okay.
Thank you so much.
See you soon. Bye.
See you soon. Bye-bye. So I guess you have like another session for the net software and then the discussion afterwards, right?
Yes, exactly. So yeah, we can keep the seminar similarly to what we did before. So I introduce the software, show something, and then yes, we discuss about certain aspects, also practical aspects, if you wish.
Sounds perfect.
Okay, great. So in this seminar, I will shortly introduce the non-invasive electrophysiology toolbox, also known as NET, which is indeed a software project that for which we have been investing the past 8 years somehow of research, because this is basically the base that we have been developing and then using for many of the research works that were somehow delivered by our research group at the KU Leuven. And basically with this software, the goal was to enable researchers to perform large-scale analysis of high-density EEG data. High-density EEG data, because we wanted to focus on it, because the goal was to extract these brain networks during resting state. Then also we expanded somehow to other kind of application, also for activity analysis, and such as ERP and ERD and ERS, to be performed not only at the sensor level, but also at the source level. So to identify truly which are the brain regions involved in a certain activity. And then also we talk about large-scale analysis, because one interesting aspect of the software is that it not only provides accurate measurements of the high-density EEG analysis, but can also be performed on many EEG data sets, because the procedure, once that the parameters are set and checked, can be run in a totally automated way. So this is something that allows saving a lot of time. Of course, there are some drawbacks in it. Means that people must be very experienced at using it, so that they can actually set the most appropriate parameters to perform the different analysis. And I'm referring both at the connectivity analysis, but also ahead on the pre-processing, so to recognize the artifacts and let's say set the correct criteria to remove the artifactual sources. However, yeah, that's something we've been working in the past years. And finally, it is available online. So there is a folder on GitHub. And now I will go more into the detail. However, the concept of the software is to allow EEG source localization in an accurate fashion, And indeed, it has been created to perform connectivity analysis, both ICA, as we saw actually from the results previously, and also C-based. Okay. And so basically, as I was saying, there is currently a publication under review for this, but already we made it open so that everyone can freely access it. So if you want to download the net also and perform your own analysis, you can follow the current link. So the GitHub and the subfolder bind group KUL, quindi KU11, and looking for the folder net. And if you want, I can show you directly this so that you can start also downloading it if you want. So I go stop share here, and I share another screen here. Okay. So now you are seeing my my computer. So I'm here in the link github.com/bind trait group trait KUL/net. And so here, basically, you can see a number of folders. Each of them corresponds to different processing steps. So we have a folder keeping inside all the scripts, the MATLAB file for performing the connectivity analysis, the data conversion, the electro detection, the head model reconstruction. So for all the different steps of the involved in the EEG source localization and analysis. And see, this is the link. Yes, great. Indeed. So you can download that one directly. So going code and then download zip. At the same time, there are still some missing information in it. And to do that, you can scroll down, for example, if you are already at the website. And there are some extra information that you can find at the osf.io link that you can open. And so basically here, you can find other information. In particular, so there is a description of the software. And also at the same time, there are some extra folders that you can see here, files. Let's go and have a look more in detail what we have here. So here we have 3 folders. One is external, template, and test data. And basically, the external contains all the functions of all the other open source software that we implemented and we used in the net. So if we go inside, you can see there is a subfolder with Brainstorm, EEG Lab, Field Trip, and some others, SPM, that are all used somehow for well, the different steps of processing. As I was mentioning before, the software net puts together some custom-made scripts, but also take advantage of the functions that are working well from other softwares. Then also you can download a template folder. And then we have a sample test data also that can be used for making some trials. There is a resting state data and also a task-related data. And here we also have the individual electrode position for the subject that performed this EEG recording, and also his individual structural MR image. And okay, so you can download also these folders. I advise you that the external one will take a long time because it has many files and many subfolders, so it is quite slow at downloading it. However, once that you have all of these in your computer, you can set up everything and let net run. So I go back to the previous presentation. There we go. Share, and I share everything here. Okay, so I don't know whether you already started downloading it. However, you can really do it also afterwards. Also on the website here, there is a tutorial that allows to approach the software, and so can be helpful for, let's say, arranging all the inputs to perform the analysis. Okay. So what we get somehow once that the file is, the folders are downloaded, is a folder called netmaster, where we have all these subfolders, the one that I was showing you before, with each of them used for a specific processing step. And so I would recommend that you also copy inside this netmaster folder your external folder, and also your template and test data folder. Well, actually, the test data can also be outside, but the external one and template should be inside this folder. And inside this folder also, you can see some scripts. More specifically, focus on net start GUI, which is in opposition to net start no GUI. This is why we decided somehow to make life simpler for other researchers, because in our research group, we were only engineers somehow, and so we all had good programming skills, and so we were working actually directly on the code. However, before releasing the software, we thought of developing also a GUI, which is actually what I'm going to show you briefly today. So in this case, you should open this script and then let it run. So in this way, it will open MATLAB. I didn't mention yesterday that it works in MATLAB, the software. At this point, you can run the toolbox and see this GUI that will be generated. I don't know whether it is big enough, whether you can see it clearly. However, just for giving a general outlook on it. So here you can define the processing directory, which is the place where you want your output to be saved. And then you can go in the box input data, where you can load your data set, and so define the folder of origin where your EEG recordings and the electrode position information and the MR image of your subject is inside, and so you can set your path. Then also here you can load your preprocessing parameters. So therefore it's the file, a template, an Excel file where you can set up your criteria for performing the different processing steps, from the pre-processing to the source localization. And then finally, you can upload another file where you have all the information about your parameters to perform the activity analysis if you have a task-related recording, or your connectivity analysis if you have a resting state recording. And you can also select whether you want to perform the ICA or the seed-based connectivity, or both. This is also an option. See another. I'm not seeing the presentation. Oh, is an isolated case, or nobody can see it?
No, we see it nicely.
We see. Okay, so maybe I try again. I don't know, to stop sharing and reshare.
Okay.
Okay. So share again. This one. Participants can now see your application. Okay, so now everyone should be able to see.
Yeah, everything's perfect. Thank you.
Okay, great. Thank you. So once that we set the origin of our data and the parameters we want to use, we can run net. So this is the box in the middle. We can select our data set that we want to analyze. We can also choose to select all the data set that we set in the previous step and that we want to analyze. And then we can perform the different steps of processing. So at first, we need to initialize the software. So at this point, we will see more in detail in the following slides. It will build up the folders, the output folders, where all the process data will be put in. And then the different steps of pre-processing will be performed, starting from the head modeling, reconstruction, and from the signal processing and the source localization. Once that the data are source localized, then we can perform again, either the activity or the connectivity analysis, and then we can perform statistics in case of group analysis if we have more data sets. So this is actually something important about the software, is that perform this analysis data set by data set. So for each individual, we will have the information about the resting state networks, for example. So they will be extracted at the individual level, and then we can perform group analysis. So this is also very relevant in case that we want correlate, for example, some connectivity metrics with some behavioral scores or some other physiological measurements, and we can do it at the individual level. And once that the analysis are run, we have a visualization tools that allows to visualize, to display the head model results. So we can visualize the structural MR image, and we can see the resulting head model with the different segmented tissues in the different compartments. We can check also for the correct co-registration of the electrode positions to the head of the subject. And then we can visualize also whether the signal processing was successful, whether we removed the artifacts correctly. So this is something that I suggest to perform somehow with the first subjects that are processed. This is a good practice somehow, not to run immediately everything, but to have a look and see whether the processing and the subsequent analysis are satisfactory. And then it's also possible to check the activity and connectivity. I see another. Is okay with me? Okay, great. So everyone is seeing everything. So going more in detail, as I was mentioning before, here you can set the processing directory where the output of the analysis will be put. Once this directory is selected, there will be 2 Excel files that will be automatically copied inside this new folder. And in this Excel file, there will be defined the information about the input, so where the EEG and electrode position and the MR image are located, and also an Excel file where the processing parameters are described. So let's say that those ones represents some template. So what we can do afterwards is to go and check the Excel file whether to check whether everything is correctly mentioned. Okay. So here again we can give as an input our information, the input data and the parameter used for the analysis. So here is what happened when we press this button, load data set. So here you can see we can choose a data set that we want to create, and we can define for each of them the EEG file name. So our EEG file that comes from the acquisition computer of the EEG. And this can be an optional file where we describe the experiment, whether it is a task-related experiment. And here we can set where the sensor position and the structural MR image are. This can be done for each data set. So when we see here choose data set, we can make data set 1, then we add the data set, and again this will be empty for data set 2, so we can fill it in. And so we can, let's say, create and arrange all our inputs. Once we do this, we can save, and so everything will be stored, and the Excel file that was created before will be updated, resuming somehow all the input information of all our data sets. At this point, also, we can check the pre-processing parameters. which includes different steps. So the first step, for example, is the conversion, which basically allows to get as an input EEG data that are acquired with different EEG systems. So this one should be specified. Let's say that NET is built to get as an input whatsoever EEG data. However, at the very beginning, it will convert each of these different data sets into a shared format. So it's just important to specify which is the input format, and then it will be translated into something that can be read by all the different functions in the software. And then here in the box view preprocessing parameters, you can see also the parameters for the head model, signal processing, and source localization. Also, here you can edit so that you can set up the correct one. So for example, in this case, the test data that it is in the folder that you can download, we have 256 recordings that was achieved with an EGI system. And so here we say that the input format is the one from EGI. For example, for brain product, you should be writing BP, so for brain product, and so the software can understand what is the input about. And also we can chunk the recording so that we can remove what we have at the very beginning, at the very end, that eventually can be affected by transitionary oscillations before, the amplifier gets to the correct regime. And also we can select the EEG channels that we want to analyze. So in this case, should be, for example, for 1 to 255. In the case of the brain product system, for example, that has 128 electrodes, would be from 1 to 127, because we would not consider the reference electrode. And then also in case that there are some external peripheral recordings, we can set and tell to the software which is the, for example, the ECG channel or the EMG channel if we are recording muscular activity. So here, more in detail for the different steps. For the conversion, we already seen a little bit. Then there is also some information about the head model, where we can describe what is the number of electrodes of the that we expect for the co-registration of the electrode position. We can define the space of reference to which we want to refer our head model somehow, so that we need to translate the MR image from the individual space into a shared common space, which commonly is the MNI space. And then we can also describe, here, for example, you can see SMRI.TPM. TPM stands for tissue probability maps. And basically here we can give the information on how many compartments we want to have for segmenting our MR image. And to each of these compartments, in this case 12, we can associate a conductivity value. And so, yeah, these ones are some parameters that can be set. The ones that you see here are the default one, and they will probably work for 90% of your analysis. Of course, it's important that you give the correct input. For example, here the template for the electrode position refers again to the EGI system, but you can set the one of the EEG system that you are using. And then here also for the source localization, there are different approaches that are available in the software. And so also here you can choose, here, for example, the source.method, and we set iloreta, but you can select many others. And also those ones are standard parameters, and some of them, they were optimized because we noticed, we tested that they led to more accurate results. Okay, so this for the pre-processing. And oh, I thought there was one for the signal. Oh no, yeah, it's afterwards. So here I show you a little bit in detail what is about. So here is the MR that is given as an input, then it is segmented. Each compartment has conductivity values. And at the same time, here we have our electrode positions that are co-registered into our MR image. And so in this way, we build the head model that somehow model the way the electrical activity flows into the brain tissues up to the electrodes where the signal, the EEG signal, is recorded. And here more in detail about the segmentation. For example, I talk about 12 tissues, so means that we are segmenting in a kind of detailed way our MR image. And so it includes many head and brain tissues. And you can see also that each of these tissues is characterized by a specific conductivity value that tells us how easy it is for the current to flow in a certain tissue. And you can see that there are large variability depending, for example, on the fact that we are considering the bone or the CSF, which is a liquid truly, and so that makes easier to the current to flow inside. These values, they are from literature. There is a growing interest in estimating individual specific values, but let's say that at this stage we are able to set, let's say, only one value for a certain compartment, and this value is equal for all the subjects, which of course it's an approximation, so it's not going to be true, but it is the best we can so far. Let's see something here in chat. So can I ask which MR template is behind the head model? Yes, the MR template is the one from the Montreal Neurological Institute. So it is, well, a very largely used one. And it is somehow the average out of 152 healthy subjects. So it is the one commonly used as a reference. This is important to be performed also because, of course, for our group analysis, we cannot put together the information from different heads. So it's needed to move into a shared template space. So then here you can see some more details about the electrode co-registration. So here we have the MR image, the electrode position, and then yes, we can check actually also with NET whether this co-registration occurs correctly, because might also happen that depending on some error in the acquisition, the reference system might be inverted. And so that we see that this part that should be in the front is actually in the occipital one. Of course, that one would be very problematic and the results would not be reliable. So it's important that this step is performed correctly. And so also you can check this with NET, whether it is co-registered in the correct way. So afterwards, for the preprocessing, we have the signal processing parameters. So here is quite small. I don't know whether you can read all the different parameters, but I will tell you shortly that you can set up the information for detecting the bad channels and removing them. You can perform the filtering of the signal. And then you can actually do also something interesting, which is actually to remove some artifacts that are specific to the MR environment. So if you decide to perform also simultaneous high-density EEG recording with fMRI, there are some specific artifacts that are involved in it, which are the gradient artifact, which is linked to the acquisition of each volume of the fMRI, and the ballistocardiographic artifact, which represent the cardiac artifact that inside the scanner, due to the magnetic field of the MR, is somehow enhanced. So it is a very large artifact that should be removed. Otherwise, well, we cannot continue with further advanced analysis. So with NET, you can also remove these artifacts if needed. Of course, if your recording is made outside of the scanner, you can set these parameters as off. So it means that this step will not be performed. And then you can go on with the traditional EEG preprocessing for removing the artifact. Actually, I'm saying traditional, but it's not truly a traditional one in the sense that we optimized this step as well. So usually, if you have experience with removing artifacts with the EEG, you commonly run ICA algorithm, and you identify some components that are artifactual and some other ones that are considered as representative of neural signal. Well, in this case, we run the ICA several times. We run it several times because every time we run the ICA, we focus on a specific artifact. And for example, we run it a first time to remove the ocular artifact, then to focus on the movement artifact, and the muscular artifact, and then the cardiac artifact. And each time, let's say that the data that we give as an input is a bit different. For example, the signal before running the ICA to focus on the movement artifact is filtered. We filter it for let pass only the lower frequencies of the signal because we expect that the movement artifact will be in those frequency band. And similarly, we do, but in the opposite way for the muscular artifact, for which we expect that the contribution of the muscle will be in the higher frequencies. Therefore, we filter the data in with a high pass filter, and then we run the ICA and we focus on those components, and some of them they will show features and spatial patterns representative of muscular artifact, while other ones they might only be beta or gamma contribution of neural origin. So this is something that it's a pipeline that was validated and was used in several studies. Actually, it has been developed for focusing on very, let's say, very difficult data sets, in the sense that a colleague of mine, Dr. Minqi Zhao, has been working for his doctorate on high-density EEG for mobile application. So in that case, we had to deal with very large artifacts that were of movement origin and muscular origin. And therefore, he optimized this pipeline, and this one was validated and showed to lead to more accurate results compared to standard pipeline where only one ICA run was performed. And so, yeah, this is a bit the signal processing. It allows to do several things somehow. And here you can see that for each of them, there are some parameters, some criteria that are set with certain values. So those ones are the criteria that allows to automatically detect the bad components, the artifactual ones. So in that case, what I would recommend is to start with a default configuration, but then to check whether the removal was successful and eventually modify. This one you can do it yourself. You can modify the value for the threshold that allows to discriminate between artifactual or non-artifactual component. In the software, no, we did not. It allows to preprocess the. I'm reading a question here whether in the software we consider also Blink ERP or microstate analysis. This one is not included in the software. However, once that you perform the preprocessing, you can actually, access your data also with other software, or you can load it in MATLAB and use your own script. So eventually you can, run the different steps, and then you bring it out from the software and you perform any other analysis you want to do with your custom scripts. Okay, so here again, this is what we have, the typical EEG recording with many artifacts. And let's say that afterwards, after these steps, we can remove these artifacts and have a clean data set. So at this point, we are able to combine this information from the EEG recording and the head model to reconstruct the activity. And then once that we are in the source space, so that we moved from the sensor space to the brain, at that point we can perform activity and connectivity analysis depending on the data set that we have. And also here we can set different parameters. We can describe whether we want to perform an ERP analysis or an ERD-ERS analysis. But focusing with the connectivity analysis, here we can set whether we want to perform an ICA connectivity or a seed-based connectivity. And for each of these methodologies, we can define our parameters. So we can say which are the frequency bands we want to focus on. And most importantly, for the seed-based, for example, we can load a seed file. So here, for example, we have template seeds, where basically we specify the coordinates in the MNI space that we want to analyze for our connectivity analysis. So, well, here somehow it shows the dual approach that we can use for setting our parameters. So we can either use the graphic user interface, so this one, and we edit everything and then we save. And so the Excel file I was telling you before that is summing up the information will be updated. Otherwise, we can simply go in the Excel file itself and we change the things there, we save it, and then since the Excel file is inside the working directory of net will be automatically updated with the new parameters that you want to set. Okay, and then once that our analyses are performed at the single subject level, at the individual level, we can also set the parameters for the statistics that we want to perform. We can set the statistical level for significancy and also perform corrections for multiple comparisons, so that we can finally achieve, let's say, the final results with statistical differences and statistically significant features, and also extract the group level maps, for example. So here I was mentioning the seed file and the triggers file that basically define the coordinates of the region of interest that we want to focus the analysis on. And in the triggers files, this, let's say, applies for the activity analysis. We can set up all the features for the markers and, I don't know, the width of our analysis window, for example, if we want to perform baseline correction. So all these features, they can be stored in this file.mat and can be loaded into net, so that the output can be also visualized. And there is another MATLAB function that allows to generate and update these file.mat, so that for each experiment that we have, let's say, our own configuration of analysis. Okay, so once that everything is set up, so that we have our data as the input and we set the parameters, we can run the software. We select the data set we want to analyze and we can perform the different steps. And once that we perform the initialization step, so this one at the very beginning, an output folder will be created And in this output folder, there will be a few subfolders. One is EEG signal that contains the processed channel data. Then there is a EEG source that contains the source reconstructed EEG data and also the output from the connectivity analysis. And then MR data that contains all the files that are generated during the creation of the head model, including the processed MR image and the electrode position and the information for the lead field matrix that is used to translate the EEG data from the sensor level to the source level. Okay, so here a little summary of which are the steps, in which order they go. So that modeling, signal processing, source localization, and then we can choose from the activity and connectivity analysis. And once that all these steps are performed at the individual level, can be performed a group analysis with the statistics. All of these staying inside the same software, and all of these performed in a fully automated way. Okay, so it's possible also, once the analyses are performed, it's possible to visualize the output of the different processing steps. And so here, for example, we can visualize the information about the head model and check for the MR image, the segmentation, and the co-registration with the electrodes. We can visualize also channel by channel the before and after artifact removal. And this one will be displayed both in terms of time course and power spectral density. And we can display them individually or together, all the channels. And then we can also visualize the activity analysis if we have activity, or the connectivity analysis, setting up different features. We can have a look at the topo plot, so the visualization at the sensor space, or we can look at the source maps, so how that activity is generated inside the brain, truly. Also, for the connectivity analysis, we can select whether it was performed ICA connectivity or a seed-based connectivity. And for both of them, we can visualize the spatial maps of the brain. And for the seed-based, we can also visualize the connectivity matrix, showing the correlations between the different seeds. Then once that everything is done, there is the quit button, and so everything can be closed, and we are happy because all of our analyses are performed. And okay, so this is the short introduction about NET. Now, if you want, I can go on showing some applications that we did, some kind of connectivity analysis that we've been performing in the recent years, for showing also the potential of NET, especially for the seed-based connectivity. I don't know whether there are questions so far, or curiosities, or well, we go directly to the end of the presentation otherwise. Uroš, maybe?
Yeah, I think we can continue on.
Yes.
Are there any questions here?
I do.
We do have some questions, but yeah, we suggest we continue on, and then we discuss in the end. Yeah.
Yes. Okay.
Thanks.
So let's say that when we first developed the approach for the seed-based connectivity analysis, well, here now it's something that is familiar to you because I already described this methodology. Somehow we are able to get the maps and also to see the correlation between different seeds. So most importantly, with the seed-based analysis, we can investigate not only which areas are involved in a certain process or are connected somehow at rest, but we can truly see not only which areas, but also how they interact to each other. And this is a very interesting thing that we can do with the high-density EEG, because we have the information also in the frequency domain. So basically, we can study how different brain areas communicate to each other and whether this communication happens in a certain frequency band or in a certain frequency. So this is what we aim to investigate at first in the default mode network, which is, let's say, the hallmark of resting state is a brain network that characterizes the resting state condition, and that is not present when an active task is performed. So we truly wanted to see how the interactions at rest in the brain occur. And so to do that, we define these typical seeds of the resting state, of the default mode network, so the medial prefrontal cortex, posterior cingulate cortex, and left and right angular gyrus. Here we can see that there is a comparison of the connectivity maps between fMRI data and high-density EEG data. So this one we already saw before. We could see that actually there is a variation in the connectivity pattern, spatial pattern, depending on the frequency band that we are using. So what we wanted to see is actually, okay, but I want to be more specific. I want to see actually the signal in this region at which frequency is communicating with a seed in another region. So here you can also see for another seed that we consider. So the posterior cingulate cortex, again, shows a specific pattern depending on the frequency band. So for example, in the theta band, the signal from the posterior cingulate cortex connects also with the medial prefrontal cortex. In alpha band, all the regions of the default mode network are connected. And for the beta and gamma, again, the right and left angular gyri are not involved in the network. But so let's see how the correlation between the signals from each of these regions varies depending not only on the frequency band, but more specifically frequency by frequency. So here, what we are seeing is basically that for each frequency from 1 Hz to 80 Hz, we have the significance of the correlation between the signals from all the regions of the default mode network. So for each row, we have the interaction, the relationship between, in this case, medial prefrontal cortex and posterior cingulate cortex, medial prefrontal cortex with left angular gyrus, right angular gyrus, and so forth. So we are checking basically how the different interaction between the different regions of this network are communicating to each other. And so we are investigating the so-called within network connectivity. So we see how the nodes of this network interact with each other in specific frequency band. Actually, frequencies. We're not looking at the frequency band. We are even more specific. And so the dots that we see here represents a significant connection between the signals coming from these regions. And so what comes up, it is basically that all the different combinations of connections of all the regions show significant relationship in the range of 9-11 Hz. So this suggests basically that all the nodes exchange information between each other in the same frequency. So the alpha band from the 9 to 11 represents a common carrier frequency that is used by all the nodes of the default mode network to exchange specific information. However, what we found out is that it might happen that different nodes exchange information also in other frequencies. And this can be done selectively for different nodes. For example, here at 14 Hz, we can see that the significant correlations affects only the relationship between the medial prefrontal cortex and all the other nodes. So this suggests that the medial prefrontal cortex preferentially communicate with all the other nodes of the default mode network at the frequency of 14 Hz. while we can see that in this frequency, this doesn't occur for the connection between other regions of the network with other regions. For example, the posterior cingulate cortex doesn't show significant connections, and so also for the right, and also between left and right angular gyres. So means that 14 Hz characterize only the connection involving the medial prefrontal cortex, while the other nodes, they don't communicate in this frequency. Analogously, we can see that for 17 Hz, for example, it is the posterior cingulate cortex that connects with all the other nodes, while the other regions, they are not connected to each other. So before medial prefrontal cortex and left and right angular gyrus, they were communicating to each other at 14 Hz, but they don't communicate at 17, while the posterior cingulate cortex does communicate at 17 Hz with the medial prefrontal and with the left and right gyrus. So let's say that with this, we can see that there is a selective communication depending on the frequency band, depending on the node involved. However, there is still a common carrier frequency at 911. So it's like that all the nodes are connected at that frequency, but all the nodes, they also communicate at other frequency depending on which kind of information they are exchanging with the other nodes. So in this context, it becomes very interesting to eventually see whether what is the meaning of this communication? I mean, why they are communicating specific regions in specific bands, in specific frequencies, and eventually correlate this also with behavioral performance, because there might be that there is a preferential exchange depending on what actually we are planning to do in our activities. So once that we check these within network connectivity, we also move on and investigated the relationship of different brain activity between each other. So not only inside a certain network, but also we check how different signals from different regions are connected to each other. In this way, with introducing the concept of between network connectivity, we could test how the nodes of different networks interact with each other. So here, I take back some of the information that I already presented before. We defined a bunch of resting state networks with some relevant regions, and then we estimated the connectivity values for investigating the interaction of each network with each other in different frequency bands. So again, the within network connectivity is represented in the main diagonal, while the between network in what we see outside of the main diagonal. And we investigated also the ratio between this within and between network connectivity. So this represents somehow the baseline of what would be the functioning of the brain in a healthy young brain. And so we can see that the connectivity within network is stronger in certain frequency bands. So the networks are more segregated in different frequency bands, and these bands varies depending on the network. So for example, we can see that for the default mode, the communication occurs mainly in the alpha band, while for the dorsal attention alpha and beta, and somatomotor for the alpha is significant, but also beta is quite high. So let's say that depending on the function of that resting state network, the connections will be different from each other. In particular, we could see that for bilateral networks, the resting state networks are strongly interconnected in the alpha and beta band for bilateral networks, and in the gamma band for lateralized networks, as you can see here from the map. And yeah, somehow what we could explore with the functional connectivity performed on high density EEG recording is that we can expand what we know from functional connectivity from fMRI. And in particular, we have the frequency content information. And so this approach and also the software net specifically can be used also to investigate how the oscillations characterize the communication within a network, within the nodes belonging to a certain network, but also can be see how the different nodes of a network communicate with the nodes belonging to other networks outside, and so allows to investigate whole brain connectivity somehow, and also to identify that this connectivity is frequency dependent. So with this one, I conclude the presentation by thanking actually all the people that we've been working at this software and all the related research in the group of Professor Dante Mantini at the KU11. And a special thank, I would say, to Dr. Gaia Taberna, that has actually played a major role in the development of the GUI also, while each of the other people, from Jessica to Minchi, Roberto, Chaing, Marco, and Ernesto, we actually, I mean, together, each of us was somehow specialized in each step. We try to do our best to optimize different steps. I would say that probably Jessica and I, we were the people that knew more about everything. So in connecting the stuffs, well, anyway, big thank to all of them, and of course, thanks for your attention. This is actually the first time somehow that I'm introducing the software to new people. So I'm glad that it has been in this nice occasion, and that this will start up also, well, hopefully a very interesting and beneficial collaboration for all of us.
Thank you, Marco. I don't think you've heard the clapping.
Excuse me?
Yeah. Thank you so much. So perhaps like just one short question, like did I miss that the information, was this software developed, was it based on certain project, or was just like the idea of Dr. Mantini and the team? So it started expanding. How is it, or do you have like the funds to keep it running?
Well, yeah, let's say that this was linked to the development of fundings that Professor Mantini got starting from his late postdoc, I would say. And so basically he dedicated so far his career as a group leader at developing approaches for performing the connectivity analysis. And so, yeah, the goal, all the people that you saw in the final slide actually has been all the PhD students and postdocs that, let's say, were in his research group starting from 2012, I would say. So little by little, I mean, he could grant some fundings, and yeah, with those ones, little by little, we could focus on the different steps. So first of all, we work on the optimization of the head model. This one was done by Dr. Marco Ganzetti and Ernesto Quarta-Morales that optimized several steps. Also, Dr. Gaia Taberna, together with the GUI, actually developed very accurate systems for detecting the electrode position. Dr. Jessica Samogina worked a lot, and also Dr. Chaing Liu and me and myself on the connectivity analysis part, both for the ICA and SID-based. And Dr. Minxi Zhao worked, he had a project for his doctorate about the mobile application of the high-density EEG. And so worked on the part of the activity analysis. Dr. Roberto Guarnieri on the real-time application for allowing, let's say, online neurofeedback using the brain network information. So each of us worked on different steps. There is still work to do somehow. on all of these fields, but let's say that we started to give a first start.
Yeah, congratulations on that. Very nice. And why I was asking this is because I've seen many scenarios in which you develop something really good, but then the funding ends, so you don't have the funds to continue on with the support nor the new versions. So something similar that happened with the Cartool, and also like not only the that you don't have new versions, but more about the ecosystem that was basically built around it. So if you have like the tutorials that are you're keeping up to date, the answers that are answered in it's
actually, Urosh, this is a big issue somehow, because I mean many of the people that I show now, they are not working anymore also on this. So now we are doing this first release of the software. Of course, I mean, it's going to be quite hard to keep the maintenance and update it with different versions. However, we thought that this was the stage where it was good to be released, because many people can benefit from it.
Yeah.
And concerning the funding, I mean, I'm not sure about how it was, but I can tell the impression that I have also now that I am applying for grants. I mean, the idea was not to propose a new software. The idea was to investigate neural communication, to let's say advance science and research in that field. Then the fact that we had to develop a software, it was a side, I mean, a collateral stuff somehow. This is also the reason why the release of the software comes after many years that we have been publishing using the software, because each study was oriented to provide new knowledge. I mean, we didn't know this thing about how the different brain areas of the default mode network could communicate to each other, for example, but now we could do it because we advanced and improved the different steps of processing. So that one is a bit, I mean, I would keep this as a, let's say, golden line to follow in my also research career. If I want to develop something, I know that I want to start anyway from a truly scientific question, and then the methodology that I can develop to achieve that can be a consequence.
Great. Perfect. I think Anna, you can unmute yourself, or if you want.
Hi. Yeah, you need to turn off them. Okay. So thank you very much for sharing. Very interesting. I have multiple questions actually, so I'm not sure how many I can ask to let others also do something like. So just one short one. Did I understand that correctly, that you need like an MRI scan individual for every participant to run the net software?
Okay, this is actually an excellent observation. Let's say that ideally, yes, in the sense that if you want to get the most accurate source localization, ideally you should have both the individual MR structural image of the subject and the individual electrode position of the subject. However, we also perform some testing and check which are the consequences of not having this information. Let's say that still, I mean, we could achieve a reasonably good accuracy. So in case that we don't have the individual information, we can use a template information. So the results will be less accurate, but still can allow to understand different things. And this is particularly relevant because, well, indeed, not every research group or hospital eventually can have this information. So yeah, it is something important to be checked. Should be further checked, I would say. However, let's say that the other steps are optimized. So for example, also the fact that the head model can be built using 12 head tissue, it is already an improvement compared to many of the head model that are currently reconstructed. Most of the people, they use templates with 6 or 5 tissues. So already modeling better these other aspects will improve the localization of your data. So yeah, it's not ideal not to have the MR of the subject and the electrode position, but still, and this already we discussed actually con Uroš about it, that for our project we will do with the template position, and still we could achieve at least, I'm still in a preliminary part of the analysis, but it should work out also.
Okay. So what kind of electrode localization did you use, and can you recommend? Like, it looked like it was a camera-based one?
Yes, actually, my colleague Gaia Taberna developed a system that is based on a camera, but more specifically, it is based on a camera mounted on a iPad. So this is actually is a system in which the operator has an iPad in his hands and with a camera, and basically has to turn around the subject that is wearing the EEG net. And by doing this, a three-dimensional avatar will be generated. And then she developed a software that allows to, let's say, isolate the electrodes from this avatar, and therefore identify the coordinates of the different electrodes. And those ones, they can also be co-registered to the MR image, the individual one. And this one can be done in a very accurate way because the geometry of the avatar will be the same of the MR image. So we are not co-registering only, let's say, a number of points equal to the number of channels, but we are co-registering a whole head to another whole head. And so this one, I mean, it is published work also. There is a associated software to it, and it has been validated. And this is actually the most accurate, fastest, and cheapest approach that is currently available for doing this kind of research. And
yeah,
so,
it sounds great. We had this discussion, our team in Berlin, several times, that how can we remove like the face from this data, and we always struggled with the ethics committee, how we can like use the nice and very precise electrode positioning, but have not like all the face data saved and stored anywhere.
yeah.
Did you solve that?
Well, so if I understood well, you don't have the electrode position, and I'm not sure actually. Can you repeat what was the issue?
Like, there's always an issue with saving like this very individual data that can like relate the EEG data or whatever you recorded data to the person. So we want to anonymize, but if you have the face recorded by camera, how can we anonymize? So do you like blur the face, or is there any step?
Well, actually, no, it is an issue that I didn't think yet about. So it's good that came up.
Okay.
I would say that well, the data could be processed in a separate stage, so you could immediately detect the position of the electrodes, and then, I mean, for the further analysis, you basically only keep the file of the position of the electrodes.
Yeah.
So that yeah, these yeah, indeed, it's actually a good point. So they are having you're having issues in Berlin with this.
Yeah, well, basically maybe it's a German thing as well, but I think it's an EU thing, that you have like to really take care about like the personal data, which is, I think, a good thing. Just we need to find solutions for our brain data stuff. Yeah.
Well, I would say in that case that, I mean, you could first process the image, and then you label them differently, and then yes, those data are stored in I think it's possible to do a kind of an anonymization step as it is done for the MR images.
Yeah, true.
Yeah.
I have more questions. Anyone else want to? No. Okay. So did you try out like the heart mode for the model, like with the eye and the muscle contributions? I think it was published lately, last 2 years or something.
So that includes in the segmentation also the muscles?
Yes.
Yes, these are included.
Have you seen it?
No. If you have it around, let's say you want to share it in the chat, would be very nice.
Yeah, okay, fair.
Yeah,
And
actually, I mean, concerning the head model also, in our research group, we have been performing some research to further optimizing it, because the compartments that we define, well, let's say that we define a compartment that includes very different parts of the head and of the brain, and we assume that everything in there is isotropic. I mean, it has the same features everywhere in all the directions. So future developments also for the head model reconstruction will try to include the information about the directionality. So by using, for example, diffusion weighted data, so that we can know actually what is the preferential direction that would be undertaken by the electrical signal. Another aspect is also to include not only the direction, but the information about the conductivity of each tissue. And actually, concerning that, we have been publishing a couple of years ago an approach that allows, starting from the MR structural image, to estimate individual voxel, bivoxel conductivity values in the brain. So this one I've been doing it with a colleague in Veneto, in Padua, Dr. Giulio Ferrazzi. And so yeah, this is actually a work where we combined diffusion weighted imaging and also structural MR imaging. And yeah, for individually, for each subject, for each voxel, we had a conductivity value. So in future, these things, they could be integrated also in the generation of the head model, so that the information is even more accurate.
Okay. And next question. So for the extraction of the band, like the frequency bands, do you always just use like the power from the whole spectrum, or did you also look into the things like the 1/f reduced from the whole spectrum, so you just have like the bandpass power, like the FOOF toolbox allows to do? You know that one? So, but my basic question is, did you just use the band power in the full spectrum?
Yes, I would say so. We included, the frequency range that we considered concerning the frequency bands analysis. At the same time, for the one for the default mode network analysis that I show in the last minutes, there actually we were doing it frequency by frequency. So it was a very fragmented way of looking at the power spectral density.
I actually really like this frequency by frequency thing. I just wondered how you corrected for multiple comparisons for this one, because like it's just looks like a lot of comparisons. So you think like, okay, there's gonna be a lot of things significant just because that's statistics. So I wondered how you solved this, because I thought I think in future to leave those very strict bands and to actually look at the brain is more leading towards something. But yeah, so I really like that.
Yeah, indeed. That approach is interesting because, we know that the frequency band concept, is a very useful one, but it's also kind of antiquated under certain aspects. So it's important somehow to explore also alternative ways of analyzing our data in the frequency domain. Yeah, concerning actually the multiple comparison, I can tell, further studies should be done, of course. I can tell that for that study, actually, what we saw, the significant connections already survived the multiple comparison. So suggest that actually simply those connections are strong enough somehow, so that might be that yeah, the communication in the brain is characterized by these kind of frequency contributions. I must say that this should be replicated in other data sets, because in that case we considered, I think, 20 subjects. I could replicate similar results, and with similar I mean that we could find that the alpha frequencies, let's say, related are shared actually by the nodes, and there is specificities in the beta frequency band for other nodes, and this one I could see it also in another data set. So let's say that yeah, it should be important to have more and more cross validations.
Yeah, cool. Yeah, I really like that. Thanks a lot. I'm done with my question.
Okay, thanks.
Thank you very much.